And welcome to another episode of The Focal Thought. My guest today is Professor David Harris. How are you?
Fine, thank you.
Good, good. Thank you very much for joining me on the show today. I'm excited to speak with you. So, David, you are a graduate of Wake Forest University in Winston-Salem, North Carolina.
Correct.
You attained your Bachelor of Degree Sciences, cum laude in Biology, Mathematics, and Psychology. You've got your Master's in Medical Sciences from Bowman Gray Medical School, and a doctorate in microbiology and immunology from Bowman Gray Medical School as well. And you were a postdoctorate fellow at the Ludwig Institute for Cancer Research in Lausanne, Switzerland. And welcome to the show today.
Thank you for the invitation.
My pleasure. So I wanted to get right into it, and we can start with stem cells because that's something that's gaining traction. If you can just talk a bit about the history of the research of stem cells and how you came into that study as well.
Sure. I think the first thing we want to start with is there's different types of stem cells, obviously. The ones that get most of the attention are the embryonic or fetal stem cells, but there also are adult stem cells. Probably 20, 25 years ago, with the discovery of embryonic stem cells by the University of Wisconsin researchers, it was shown that those stem cells had the ability to become every other tissue in the body. And for that reason, we call them pluripotent or totipotent stem cells. And the idea was everybody then would be able to have a source of stem cells when they needed to have something fixed or repaired, or they needed to transplant, and these cells would be universal. It quickly became obvious that there were some ethical and moral issues with the use of embryonic stem cells, along with the fact that unless somebody had the foresight to harvest those at the time of birth, nobody really had access to those cells because when they're born, the cells go away. So they really weren't clinically applicable because you couldn't use somebody else's embryonic stem cells in your body. So we turned to looking at other sources of stem cells. And back in the 90s, in sort of our naivete at the time, we were working on stem cells and transplantation. We thought that the best alternative to these fetal stem cells would be the neonatal stem cells that are found in cord blood, because a few seconds ago those stem cells were actually embryonic or fetal stem cells. Once the child is born, the leftover blood is now neonatal stem cells, and we thought it would lie somewhere between the embryonic and the adult stem cells that you would find in bone marrow. And so we embarked on a decades-long investigation into whether we could use those cells not only to transplant people who had cancer and needed treatment, but also whether they would be applicable to things like tissue engineering and regenerative medicine. So they would be the best of both worlds. And so although people try to divide the stem cells into embryonic versus adult, we actually think there's a third category, and that's the neonatal stem cells that you find in the umbilical cord blood. And those have been shown to possess properties of both types of stem cells, and to really have the ability to have everyone collect and bank those for future use, or to have them put away for somebody else to potentially use at a fairly reasonable cost. And then the downstream applications seem to be almost unlimited.
Got it. And with the stem cells, they're essentially the body's raw materials, right, from which other cells with functions are generated?
So they're the building blocks, and how much building you can do with those blocks depends on when you harvest those cells. So the ones, as we talked about, the embryonic can become everything. The ones that would be found in an organ like the liver or the kidney can only become those tissues. And then you have the others that are in between, like those in cord blood, that can become 5 or 6 different lineages of cells, but not every cell in the body. And so you really have to be careful when you talk about where those stem cells come from as to what you could potentially use them for.
Correct. And in terms of usage, it's basically understanding about diseases, regenerating healthy cells, testing for new drugs, safety, effectiveness, things like that.
A variety of applications that we would normally use cells and tissues for. So in sort of the personalized medicine era that we're in currently, you would like to know if your body, not just the average of everyone's body, would respond favorably to a new drug or a new therapy when you need treatment. So you could either use tissues that were derived from a bunch of donors in the past and hope they apply to you, or potentially you could use the stem cells from your body, make them into that tissue, and then see if your own particular ones respond favorably. And that's really where we're going in terms of the use of stem cells in personalized medicine. And knowing that information, you can then direct those stem cells for regenerative medicine to repair tissues and organs, or tissue engineering to make, say, a heart valve, or even to do things like transplantation for patients who have leukemias or other types of cancer.
Got it. Okay. And then you mentioned earlier about, the ethical and some of the controversial issues with stem cells. And these are, I imagine, are the embryonic stem cells. So if you just talk a little bit about that, please.
This was more a political issue than a scientific issue, as things often are these days in the scientific realm. So the idea was that embryonic stem cells, that is, that the leftover embryos from in vitro fertilization, were shown to possess these stem cells that could become every cell in the body. And so the idea was, or the political idea was, then that was going to encourage people to have elective abortions in order to get fetal stem cells. It ignored the scientific fact that those stem cells can only be harvested during the first 7 days of fertilization. So there's no embryo there, there's no child there, there's nothing to abort. And they're always made in the laboratory. You can't actually take aborted fetal tissue and use it to make stem cells. But as I think we're all aware these days with the other issues, that was blown out of proportion. But that wasn't the reason for those stem cells not making it to the clinic. The reason was you could not harvest those for an individual, and you couldn't use somebody else's fetal stem cells for your own particular needs. So you needed to have autologous or your own stem cells to be able to do these treatments, rather than the use of the embryonic. So the embryonic stem cells really have gone into mostly drug testing and sort of mechanism investigations, rather than clinical applications.
Got it. Okay. And, with the success of stem cells, we've heard about, celebrities such as Mel Gibson, who had his father, I believe, undergo some stem cell operations. And then even Joe Rogan as well. I mean, they talked about it on that podcast. So there are certain areas or countries that, the operations are not allowed, but then there are certain countries and, places such as Panama where they are allowed, right?
Correct.
What's the deciding factor between that? Is it political, as you mentioned, primarily, or?
Well, you have to be careful, with anecdotal reports, with one-offs, particularly with people who pay tremendous amounts of money to have these procedures. It behooves them to say the procedures were successful, so they don't appear to be scammed or have undergone something that was not worthwhile. So there are places both in the United States as well as outside of the country where you can have certain types of stem cell therapies performed. Those tend to be those, at least in the United States, they tend to be the therapies that have some proven benefit. For example, to treat osteoarthritis of the knee or the other joints, or to treat stroke. But then there are other procedures which are more questionable, that are performed generally outside of the United States or outside of the country with less regulation, where I would be concerned that there might be some side effects or some other problems that could cause more harm than good. So we want to be sure that when we talk about doing regenerative medicine using stem cells, we either talk about it with the caveat that it's either unproven or under investigation, or with the restriction that we know it works for this, but not for that. So let's don't make an assumption it works for everything.
Got it. Yes. And, from what I have read, there are some problems with the embryonic stem cells in humans. For example, if, you're using the stem cell to, become a certain type of cell, say, for example, to become heart cells, these cells, in terms of growing irregularly or, growing spontaneously, learning how to control it and, differentiating the embryonic stem cells, that's also undergoing in the research, right?
Exactly. There's always the problem that these embryonic stem cells can become everything. And when you place them into the body, they're not really sure what they're supposed to become. So they do become everything. And then you end up getting either benign or even malignant tumors forming at the site of injection, which is not a good thing to happen if it's inside your brain or inside your heart. You don't want that to occur. So you have to be very, very cautious about those kind of investigations, those kind of treatments. to make sure that there is support not only for the assumption that it would work, but why it would work, and there should be at least some preclinical data to support the potential success when you move from the laboratory into the clinic.
Got it. And from your experience in the work that you're doing, where are we in the timeline of this research? Are we still sort of in the early stages, or are we starting to get a grasp on it? Or
I think we are starting to get a grasp on certain types of applications. I think one of the ones that seems to be particularly successful is the treatment of osteoarthritis, the joint and knee and back and hip degeneration using stem cells. Our own experience, as well as those that I've seen published and in the clinics, shows that seems to work very, very well. That doesn't mean you can now jump over doing the treatment of the osteoarthritis and start to make ligaments and tendons, but I mean, at least at this point, that the osteoarthritis seems to be fairly well established, and there's a reasonable step to get to the next stage. However, that's a big jump from, say, treating somebody who's had a heart attack or a stroke using stem cells, where there's less preclinical work done, and so there's less evidence as to what's going to be successful down the road. So that works well. I've seen stem cells work well for stroke in children in clinical trials. I've seen it work well for inflammatory diseases, like autoimmune diseases, where it works well. But then I've seen that it has not worked at all for other types of applications. So again, we don't want to assume or make the assumption that stem cells are the holy grail, and once you have them, they'll cure everything. That's not going to be true. But then again, it's the same as also we can't say it's just a scam or pie in the sky hope. There are things that it appears to work very well for, and there are other things that it doesn't appear to work at all for.
Got it. No, that makes sense. And a branch of stem cells is therapeutic cloning, right?
Well, it's a branch of that research. I think you would have a hard time getting people to agree that they want to be associated with therapeutic cloning. Regenerative medicine might be a better way to word that.
Sure. What are your thoughts on therapeutic cloning or regenerative medicine?
Well, regenerative medicine seems to have tremendous potential and has started to produce some very good clinical results. Again, the therapeutic cloning tends to be associated with things like, making new people or cloning yourself. So we try to keep that at arm's length. If you want to go out and try to take stem cells and make yourself a new liver or a new kidney, that's reasonable, although it's unlikely to be successful. It's more likely to take the stem cells and place it into the damaged liver or kidney to help it repair itself. And although that's not typically classified as cloning, it is regenerative medicine and has a much greater chance of success than trying to grow a whole organ and place it into the body.
Got it. Yeah. And who knows? I mean, with, a bit of creative imagination and science fiction, we might look into, with these stem cells research going on, creating a brand new organ, possibly, right, for the human body in the future.
There is work on that in livers and kidneys, where they'll take your liver or your kidney out. They'll decellularize it to get rid of all the damaged or diseased tissue, and then try to reconstitute it using just the infrastructure or the frame of that organ, and place new stem cells in it and grow that new organ and put it back into you. It seems fairly straightforward and simple on the surface, but you have to realize that each organ or tissue has a multitude of different cell types that do a whole bunch of different functions, and to get all of them grown in the right place at the right time and interact with each other is not quite as simple as it seems. So it's more than simply placing an empty frame and putting cells on it. You're going to have to put a whole bunch of different frames, a whole bunch of different cells at different times, and get them to talk to each other. It should be feasible, but it's probably not in our lifetime, unfortunately. What is in our lifetime is during the early stages or the early window of the tissue or organ being damaged, we may be able to reverse that by placing stem cells into the organ, so that all they have to do is fix the small part that's damaged or dying, rather than try to replace the entire organ.
Got it. Just a portion of it rather than the whole part.
Right. Yeah.
Got it. Okay. No, that's great. I wanted to switch gears a little bit here. I wanted to talk to you about microbiology and immunology,
Sure.
which you're well versed in. Immunology is something that's really important. From the work of Edward Jenner, I believe it was the 18th century.
Yep.
Where his work led to vaccinations and things like that. Humans as a species, not only do we have threats from the forces of nature, or maybe even other species, but there are also threats from microbes and our own immune system, reacting against plants, as we see in allergy systems and things like that. When humans are born, are they born with a proper set of governing principles for an immune system? I know some do have autoimmune deficiencies and things like that, but for the most part, are we born with something that's ready to go?
For the most part. So if you look uh, back at the work that was done to try to fix genetic diseases in utero by transplanting cells while the fetus was still in the mother's womb before it was born, we see that very early on during the pregnancy that the immune system is competent and can reject cells from somebody else when you try to do these therapies. So the immune system is formed early on. When the child is born, there is a functional immune system, but it's not a mature or complete immune system. It waits for the interaction with the environment to set that up. And so when you look at the children who are born to mothers vaginally versus cesarean section, or children who breastfeed versus those that are formula fed, those that encounter the natural microbes out in the environment, either through the vaginal delivery or through the breast milk, tend to have fewer allergies and fewer overreactions of the immune system, because they're learning early on what's good and what's bad, what's okay to keep around, what's not. And so that there is a flexibility at the time of birth that lasts for about 6 to 9 months, where the immune system is learning what's foreign and what's not. And obviously, we want it to react to the foreign stuff, like the viruses that come around, but we don't want it to react to our stuff, like the normal skin microbes that do good for us. So it is functional. It's just not completely matured and fine-tuned at the time of birth, and it does take about 6 to 9 months. And that is one of the reasons we actually encourage, breastfeeding. We encourage having dogs or cats around as pets, because those kids tend to have fewer allergies and fewer asthmas than those who live in a sterile environment. You got to remember that every time we walk around in the house or outside, we're encountering hundreds of thousands of new microbes at any time. Take a breath, pick something up. We don't want to react to those because they're not harmful. We want to be able to determine the needle in the haystack that's a problem and react to that. And so we have to educate the system during that first 6 to 9 months after birth, so that it knows what's good and what's not.
Got it. And it's quite a delicate balance, because from what you're saying, you want to, have the child play in the puddle of mud, but you don't want the child to, garner any diseases from the puddle of mud, right?
Right. And so we would encourage the kid to eat from the puddle, the puddle of mud, because it's unlikely that they will, encounter a life-threatening organism by doing that. But then again, we want the immune system to be ready just in case. So, when you look at it, luckily most people don't have autoimmune disorders or immune deficiencies. It's fairly rare. There's a lot of them, and so when you look at the total number of people, it appears to be a lot. But on a percentage-wise, it's less than a percent of people have these issues. So the immune system is tremendously successful at what it does, in terms of being able to protect us from the outside world or the dangerous part of the outside world, while keeping the good part of the outside world happy.
Got it. Okay. And you mentioned that the window is, I believe the first 6 months for it to learn. What happens after that? Does it stop ad hoc or abruptly?
There's not as much flexibility after that 6 to 9 months. It's been educated. It's more or less set up its path. So if the immune system's been kept in a real sterile environment, and this is from our own experience and those of our friends, when you had that first child, every time they dropped their pacifier or whatever, you boiled it, stuck it back in their mouth. And by the time you had your third or fourth kid, you just kicked it back over to them, because you realize it actually was better for them if they put some of that in their mouths. And so, um, yeah, I think we want to be cognizant that there are things out there that could harm the newborn, but for the most part, we want it to interact with the animals, the pets, the plants, etc., so that they don't grow up in a sterile environment where they don't know what's a pathogen or what's bad, and what's actually something that is helpful or good.
Got it. And by this sentiment, the 5-second rule might get extended now to the 10-second rule. So
I think for young kids, maybe for older adults as we get older, it's not, but for young kids, yeah, it's perfectly fine for the vast majority of cases.
sure. And just in a very basic way, if you could explain, what is the science between, if it's too sterile, it's not good for you, whereas you need some sort of familiarity and introduction with some of these pathogens. Why is it that way?
Right. So remember, during the gestation in utero, you're kept inside the placenta in this sort of semi-sterile environment where you're getting the nutrients and exposure to the mom so that you can grow and develop. And then all of a sudden you're placed outside into the real world, which is full of a bunch of stuff you've never seen. And so you're trying to tell the immune system at that point of where it should make a reaction and be aware, and where it should not. And so it's at that point during that 6 to 9 months that it is learning. So if for some chance you don't make a response to a potentially harmful pathogen and it starts to cause an inflammatory reaction and make you sick, the immune system will eventually kick in. And if it doesn't, thank goodness, these days we also have antibiotics that we can fortify it with.
Yes. And
so I think, at that point, we want to say, the reason that you and I have a really good chance of living till, our 80s versus turn of the century where it was more like in the 50s is because we have safe food and water for the most part, so it's free of pathogens. We have antibiotics just in case it's not, and we have vaccines to protect against diseases that spread too rapidly for our immune system to respond and keep us healthy. And so those three things over the past 100 years have almost doubled our lifespan.
got it. No, for sure. And I remember, I forget where it was that I was reading that not too long ago, people were actually in some parts of the world drinking beer, as opposed to water, because water wasn't safe to drink, but beer was.
Right, during the Middle Ages. Of
Yeah.
course, this is not alcoholic beer. It's like, the non-alcoholic beer you buy today with O'Doul's or whatever. But the reason was to make beer, you had to boil the water. And boiling the water sterilized it from the harmful pathogens, and also then provided calories. And so, that's that show on TV, How Beer Saved the World. And it actually has to do with the fact you have to boil the water, which is really the important step there.
Got it. No, for sure. And you had mentioned vaccines, and it seems that that's the popular word, especially in times like today. But we've seen vaccines come out that have reportedly prevented the spread, like you mentioned, of many diseases. We're looking at polio that has come down to, I think, about 2 countries now. With some of the work that's been done, smallpox, many influenzas, things like that. And there are different types of vaccines. One where they actually administer the actual, viral dose, as opposed to now where they're sort of, modifying the DNA with the mRNA vaccines, right?
So it really depends on the particular pathogen, whether you need to administer a killed version of it so it can't make you sick, or a non-pathogenic version of it so it goes through 1 or 2 cycles of infection and then dies off. Or if it's well characterized, like the recent COVID viruses, where we can actually take the DNA out and use that to make the antigen or the protein that stimulates the immune system. And so the idea was for certain types of vaccines where we really don't know what part of it causes the immune response, you like to use the whole virus or the whole pathogen. For others where it's very well characterized, like influenza, we can just isolate the proteins and immunize you with the proteins to get the same immune response. But the idea is that by giving this leg up on the response, you won't get sick and die before your immune system can kick in. And so you have to remember a lot of these viruses and bacteria can duplicate or double themselves, in a matter of minutes to hours. And our immune system, the first time it sees a pathogen, generally takes 5 to 7 days. And so if you don't build in that little buffer to give it time to kick in, the pathogen or the microbe will overwhelm you before you can make a response. And so that's the idea behind the vaccination, is before you can succumb to that microbe, we're going to give you a little bit of buffer to make sure. Now, the vaccines by themselves then work in combination with the immune system when it kicks in, to clear you of that infection.
Got it. So is it that the vaccine itself is not curing you, but like you're saying, it works with the immune system? So really it's the immune system that's, preventing it. So the vaccine sort of giving it a head start.
Yeah, the vaccine primes the immune system, so it's already seen it once. It's gone through that 5 to 7 day period, so it's ready. So the next time that it sees the infection, it only takes 1 to 2 days. And so in 1 to 2 days, it can quickly respond and get rid of the underlying infection before it spreads, before it gets too big. So the vaccine is priming or boosting the immune system so that it's ready to go the next time you encounter that microbe.
Got it. So, we were talking about the vaccines that administer a small sample of the pathogen, but then now with the mRNAs, it's actually, ordering the DNA so that when it recognizes that pathogen, only at that point to go into defense mode. Is there a benefit you find with one or the other, or one that works better, or does it depend on the pathogen?
If you know what the protein is that causes the immune response that you need to make a response to get protection, using the genomic or the DNA vaccines or the RNA vaccines is really the easiest way to go. There's not a lot of extraneous proteins you have to worry about that might dampen the immune response or cause a side effect. If that protein, like the COVID one, alters itself a little bit, you can quickly switch in and out those genes. You can make them in much larger numbers for a much cheaper cost, quicker, so that year to year you can quickly change these to make sure they're tailored exactly to what's needed. I don't think people realize that we have to start making the influenza vaccine 3 to 4 months before flu gets here. And we do that by monitoring what type of flu is present in Asia as it makes its way around the world each year. And there are times when by the time it gets here 3 to 4 months later, it's changed and we got the wrong vaccine. This way, with the genomic vaccines, we can actually wait until a couple of weeks before it gets here and then slip that piece of DNA in, quickly grow up the vaccine and have it available. So it's going to allow us to have more finely tuned vaccines at cheaper cost. It probably will allow us to also develop vaccines against things like cancer that we don't currently have, using current technology.
Got it. And what, if I could ask you, what might you advise or say to people, especially these days, who are apprehensive about the COVID vaccine? And once again, I don't blame them, because they don't have based on some past history, they don't have trust or faith in their leaders, or the politicians, or
right.
in the media, things like that. But they themselves are not doctors. And even some doctors out there, they mean well, but again, unless you're looking at the virus in a microscope and you really know your work, that's also subjective. So what would you tell these people who are a little bit apprehensive on taking the vaccine?
Well, it's not just this particular vaccine. If people are hesitant about taking any vaccine, again, I would point out the fact that you're going to live to 70 or 80 is because of vaccines. It's not because of anything else. When you look at the current vaccine, politics got involved more than science. And so, by the time that we had administered, 180 million doses in the U.S., and by the time we'd administered close to 2 billion doses worldwide, there's not really much more to wait for in terms of seeing what's going to happen. People say that they're afraid of the side effects, but when you look at the side effects, it's the same kind of side effects you see with the flu vaccine every year, or with the MMR vaccine or the shingles vaccine. It's actually a good side effect, that you get a little bit of fever, you get a little muscle aches, you get a little bit of chill, all of which indicates the immune system has been kicked on and is working. I'm more concerned when after 1 or 2 shots, I don't feel any different, that the immune system hasn't recognized that and is not making much of a response. If you look at the incidence of significant side effects, and again, you have to look at the literature, not Facebook or not YouTube. If you look at the incidence of significant side effects, it's less than 0.001%. So you've got a 99.99% chance of having a good outcome, and I would take those odds to Vegas anytime.
Got it, got it. And so with vaccines, there will be side effects, like you said, no matter what the type of vaccine, and sometimes that side effect might cause more harm than the vaccine was intended to do. But it's
extremely rare, extremely rare. When you look at, the coronavirus, this particular COVID infection has a mortality of close to 2%. Close to 2 people out of 100 who get infected will die. Of those who get infected and have a positive outcome, about 20 to 25% of those have what's called long COVID, with significant, cardiovascular and neurological side effects for the rest of their lives. When you look at the number of people, and we saw this as first responders, the number of people who've had, really bad outcomes, having a little bit of aches or fever or chills from the vaccine seems to be a pretty small price to pay. And so again, I'll take the 99.99% chance of good outcomes without any side effects with protection down the road anytime.
No, that's great. And once again, thank you again for, coming on the episode and sharing your experience with us. If people want to learn more about the work that you do, or, look at your publications, where's the best place to reach you for that?
Probably the best place is to go to the University of Arizona website, the www.arizona.edu. And there you can, in the little search bar, you can put my name in and it'll pop up my affiliations and my contact information. So happy to answer any questions or provide any sort of information that somebody might think is useful.
No, that's great. And I'll put that in the show notes as well.
Okay.
Okay. And thanks again, and I wish you well.
Thank you. Have a good weekend.